Live natin.
Kino-watch lang po sa
So as
YouTube, sir.
always, these lectures are recorded, and if you want to come back to them, feel free to do so. Patrick will give you a link later. Hindi ko alam kung may mga GC kayo dyan or whatever. Basta, alam ko, I am 100% sure ibibigay yan ni Patrick. Di ba, Patrick? Anyways, let's continue with our discussion. First on the list is Staphylococcus and Micrococcus, or the family Staphylococci and Micrococci. Okay, so we go to our first slide. Hang on a sec. Ba't ako ang nawala? Ako naman ang walang ano. Ayan, nakita ko na. Minimize ko pala kayo, sorry. So our first topic is Micrococcus and Staphylococcus. Staphylococcus, Micrococcus. We need to understand what causes confusion to many people who have been studying MLS. Many MLS students who've been studying microbiology. And one of the things that causes them confusion is the family Micrococci. So there's 4 species that you have to remember, and this includes Staphylococcus, Tomatococcus, Micrococcus, and Planococcus. We are not going to discuss Tomatococcus and Planococcus because they are more or less for vet techs. Hindi tayo vet techs, hindi tayo mga nag-handle ng animals class. So, huwag kayo masyadong stressed out. Okay? Pag lumabas yan sa exams, ignore nyo lang siya. Okay? So, what we're mainly focused on is bullet 2 and bullet 3. Okay? So, Staphylococcus, Micrococcus. Okay? So, under the family of Micrococcus, or when we stain these two things, that's our next slide. When we stain these two organisms, they would have different morphological characteristics. We always go back to the morphology. Tingnan nyo, every time na magdi-discuss tayo ng microorganisms, gram stain ang una lagi nating tinitingnan. Because Staphylococcus, they have a characteristic cluster appearance or grape-like, hence the term staphylo. Okay? Micrococcus, on the other hand, they come in at cocci in tetrads. Okay? So para siyang, alam niyo sa Tetris, yung box doon sa Tetris. I don't know if you guys are young enough to remember what Tetris is, but in Tetris, we get tetrads. So tetra from the from the Latin word tetra, 4. Okay. Now, one thing that causes confusion to many people. Next slide, Patrick, is saan ako? Sarcina species. Okay, kanina sinabi ko na sa inyo yung Sarcina species. They can be identified from other members of the Micrococci family because they appear in tetrads of 8. Di ba? Naalala nyo si Gaffica tetragina at saka si Gaffica tetragina and Sarcina lutea. Okay, so they appear in tetrads of 8. Ayan. This is from my professor. Di ba? Hindi ko alam kung saan kinuha ng professor ko. Shout out to Sir Mortel. Okay? So, Sarcina lutea. This is the Sarcina species. Nakita nyo, 8 packs of tetrads sila. 5 packets of 8 sila. 8 packs of tetrads tuloy. Packets of 8. Okay? And they have a characteristic growth pattern on blood augers, on blood auger plates. They usually appear creamy white, pinhead colony. They usually appear as creamy white pinhead colonies on blood auger plates with a characteristic hemolytic pattern based on species. Okay? So, they have a characteristic pattern. What does NB stand for? That is novobiosine. Mamaya natin pag-usapan yan. Okay? General characteristics pa lang tayo, class. Okay? So, pinhead. Iba yung pinpoint pala, guys. Pinhead colonies, creamy white, with a characteristic hemolytic pattern based on species. And because we're talking about hemolytic patterns, what are the 3 types of hemolytic patterns? Pupunta tayo sa klase. Claire Gono. What are the 3 types of hemolytic patterns? Ganun, di ba? Yung mabibigla, yung maninindak si Sir Manuel. Bigla na lang mananawag ng tao. Ayoko na nagre-raise ng hand. Ako naman ngayon ang magtatawag. What are the 3 types of hemolytic patterns? Wala. Next person tayo. Maria Therese. Wala na, hindi na ako magtatawag.
Alpha, beta,
Ano, te?
tsaka non-hemolytic. Alpha, beta, tsaka non-hemolytic po.
Okay, very good. Or gamma hemolysis. Okay? Sorry, hindi pala ako na. Okay, hindi ko naririnig si ano. Okay. Hindi pala ako naka-headset. Okay, so incomplete hemolysis is alpha hemolysis. Beta hemolysis is complete hemolysis characterized by the green zone for alpha, clear zone for beta, and no hemolysis for gamma hemolysis. Okay? Now, let's move on to the next one. This is your slide. This is the picture. Okay, let's move on to the next one. So, pupunta tayo sa alpha hemolysis muna. What is produced by alpha hemolytic organism as a result of the characteristic color of blood agar on the blood agar plates? So, anong na meron sa blood agar plates? Bakit naging green? Methemoglobin. Okay? MHB. Okay? Methemoglobin. Okay? Now, we're going to talk about a specialized hemolytic pattern or a special hemolytic pattern that can only be that can only be seen by certain strains of staph species or strep species. And this is known as this is characterized by incomplete hemolysis with an inner zone of complete hemolysis. This is known as alpha prime hemolysis. Look at the difference between the two. So nakikita nyo, medyo yellowish yung si left side, right? Kita nyo? This is, by the way, credits to Microbe Online. Green, yung medyo greenish na side or yung may yellow-green tinge, yan yung classic alpha hemolytic pattern. Okay, the one on the right is the same thing, but near the colonies, yung colony natin yung parang little slit na yan. Makikita nyo, complete hemolysis yung gitna, tapos yung nasa labas, may hemolysis pa rin. Almost the same color as the one on the left, yung the outer periphery ng slide na, yung picture natin na to. So nakita nyo, so alpha prime. So pag sinabi natin alpha prime, there's something near the colonies. Okay? So complete hemolysis in the colonies ang ibig sabihin nun. Ay, ang meron doon yun yung something na yun. Okay? Hindi ko pala dinapat sa akin yung camera. Okay? Clear? Clear ba sa atin ang difference ng alpha hemolysis and alpha prime hemolysis? Uulitin natin. It is characterized by complete hemolysis with an inner zone of complete hemolysis. Sir, ba't hindi na lang tinawag na ano? Pinapahirapan kami. Lumalabas tuloy yung pagka-Ilongga ko kahit hindi naman ako Ilongga. Bakit ganon? Diba? Kasi, dalawang klase kasi usually it causes confusion to many people. So, binigyan na lang nila ng name. Okay? Binigyan nila ng name. Alright. So, let's move on to the next slide, which is, what were we discussing with this one? I think this has something to do with Staph aureus. Oh, yeah. Okay. So, Staph aureus is characterized by what type of hemolytic pattern on blood agar plates? This is beta hemolysis. This organism is beta hemolysis. But this organism demonstrates beta hemolysis on blood agar plates. And there are 3 species of organisms under the Micrococciae family, which is under the genera of Staph or of Staph, that must be differentiated also from Staph aureus. We have Staph citrus and Staph albus. They can be differentiated using Loeffler's serum slant. I think that's the next slide. Oh yeah, okay. So he showed that. On Loeffler's serum slant, you see Staph aureus as golden yellow, Staph citrus as lemon yellow, and Staph albus as porcelain white. Look at the difference between the names. Okay, aureus is golden yellow. Citrus, ano bang color ng citrus fluids? Ng citrus fruits? Lemon, di ba? Citrus, lemon. Tandaan nyo lang anong naaalala nyo pag citrus fruits, lemon. Anong color ng balbas ni Albus Dumbledore sa Harry Potter? White. Porcelain white, di ba? O para hindi na kayo mahirapan. Ayoko kayong nahihirapan, class. Okay? Anong color ng balbas ni Albus Dumbledore? Porcelain white, di ba? Yun yung first description nung nakita ni Harry Potter si Albus Dumbledore. A stout old a gingerly old man with an aura of whatever, and then porcelain white beard. Diba? Mga ganon. Diba? Para hindi nyo nakakalimutan. I want you guys to make neural connections with these things because these information that I'm giving to you, they need they need to stick to your brains. Okay? Kailangan hindi siya mawala. Kailangan maitato siya. Okay? Anyways, let's continue. Okay? Masyado na akong masyadong intense. Sir, you need to chill. Sir, you need to chill. Okay, so this is considered as a selective differential media for Staph aureus and other medically important staph. So there's three organisms that we can differentiate using this type of media, the selective differential media. And what I'm talking about is mannitol salt agar. Okay, so mannitol salt agar has an indicator known as phenol red. And the sugar that is there is, of course, mannitol. So mannitol salt agar. Staph aureus has a yellow colored colonies, while Staph epidermidis and Staph saprophyticus has a pink colored colony when grown in this culture media. Okay? So para hindi kami nahihirapan sa ospital niyan kapag nilagay na sa MSA, specifically kapag yung mga pasyente may cystic fibrosis, ginagamitan namin ng MSA yung mga pasyente. We call it MANS and SABS. Okay? Bakit? Kasi kailangan namin malaman kung may Staph aureus yung plema ng pasyente. Kaya ginagamitan namin ng SABS and ng MANS agar yung samples that comes from patients with diagnosed cystic fibrosis. Kasi nga baka MRSA yun, hindi siya magamot. Anyways, let's move on. What is the inhibiting substance used in MSA? So, salt siya dapat, di ba? So, mannitol salt agar. So, may sugar siya, mannitol na yon. Yung salt, yun yung inhibitor. How much salt is in mannitol salt agar, by the way? Next slide na tayo. So, that is the inhibiting substance in mannitol salt agar is 7.5% sodium chloride or table salt, di ba? Sodium chloride. Alright, let's move on to the next slide. What is the test used to differentiate non-mannitol fermenting Staphylococcus species? Balik tayo doon sa, mamaya tayo mag-move forward. Balik tayo sa 5 slides before, Patrick. Isa pa. Dalawa pa. Sobra pala ako. Sige pa. Up. We'll see it when I see it. Now, up. There we go. So, yan yung NB. Okay? That is how we differentiate other staph species, other beta, non-beta hemolytic species of staph with each other. Ano yung NB na yan? Sino makakasagot niyan? Ano yung NB na yan? Rochelle. Ronchbel pala, sorry. Ronchbel, hindi pala Rochelle. Sorry. Ronchbel.
Sir, Novobiocin po.
Novobiocin. Okay, sorry. Kaya pala hindi siya nagsalita kasi Rochelle ang hinahanap ko. Mali pala ang basa ko sa name. Sorry naman. Novobiocin. Very good. Balik na tayo dun sa kanina, Patrick. Naloka ako kay Ronchbel. Ang hirap naman i-pronounce. Ano bang apelyido ni Ronchbel na lang? Apelyido na lang tayo. Very formal. Comedor na lang tayo. Okay? So, Novobiocin test or Novobiocin sensitivity test. Novobiocin sensitive, we will have Staph epidermidis, and Novobiocin resistant is Staph saprophyticus. Okay? Staph epidermidis sensitive or susceptible, Staph saprophyticus resistant. Okay? Clear? Clear tayo? Okay? So when you see something that has when you look at something on the gram stain, it has clusters that look like grapes. What do you have? Micrococciae or staph? Staph. How do we differentiate Staph aureus from other non-beta hemolytic staph? If for example it doesn't look as beta hemolytic as it should, because in some specimens it would look the beta hemolysis would not be as apparent as in some other specimens. You would do Loeffler serum slant, diba? para ma-identify siya from which other organisms? Staph albus and Staph citrus. Diba? If, for example, hirap na hirap ka pa rin, you use MSA. What is MSA? Mannitol salt agar. There are 3 types of organisms that are differentiated, or three types of medically important staph species that are differentiated using mannitol salt agar. This includes Staph aureus, Staph saprophyticus, and Staph epidermidis. Yellow colonies will be for Staph aureus, di ba? Recap lang ako para hindi kayo mahirapan. Ngayon, for example, you grouping colonies. What is the next step? You use novobiocin sensitivity. Which one is sensitive? Sino nga ang sensitive, class? Pakitype nga kung nakikinig kayo, class. Which one is sensitive? Naku, walang nagtatay. Patay tayo. Complete pati class tayo ako sa sagot. Ano la? Ayan na nga ba? Meron na nga sumagot. Si Jericho. Si Jericho, may sumagot na. Complete tayo. Gusto ko kumpletuhin ninyo, class. Okay? Baka mahirapan kayo sa exam. Okay? Staph saprophyticus ang what? Resistant, hindi sensitive. Okay? Ayan na nga. Oh, balik mo nga doon. Ano? Balik mo kay. Ano? Balik mo, Patrick. Ayan. Pag sinabi ko sensitive, susceptible, it's the same thing, class, ha? Baka nahirapan kayong ano. Okay? Staph saprophyticus resistant. Okay? Okay, clear? Clear? Okay. Now, how do we differentiate staph from strep? Sige nga. Doon sa mga nag-work sa ospital. What test do we use? Gagram stain natin. Hindi siya mukhang clusters. Nalilito ako, sir. Anong sasabihin ng staff mo? Gumawa ka ng ganung test. Kumuha ka ng dugo ng rabbit. I-centrifuge mo ang dugo ng rabbit. Nasa citrate nasa citrate. Oh, anong test yon? Kayo talaga. Marion.
Sir, catalase test po.
Oh, okay. Tama, catalase. Kala ko malito ka sa sa coagulase. Very good. So catalase test has what? Reagent 3% hydrogen peroxide, and the positive result is effervescence. Ano yung sinasabi mong effervescence, Sir Manuel? Ano yung kaartehan na yan? Hoy, lumabas yan sa ASCPI na test bank. Okay? Nung bago ako, ano yung effervescence na yan? Dinictionary ko pa yung class. True story, dinictionary ko pa. Ibig sabihin yung effervescence, nagkakaroon ng bula. Okay? Yung parang pag nagbukas ka ng ng bote ng can ng Sprite, tapos lumabas yung bula na ganon. Sprite ang ginamit ko, hindi Coke. Okay? Copious bubbles. Ang arte, di ba? Marian Venice. Okay? So akala ko malilito ko si Moyo ng coagulase. Catalase ang sinagot niya. Very good. Correct. Okay, correct. Alright, let's move on to the next. This is the answer. Catalase test, reagent, 3% hydrogen peroxide, H2O2. Sige, kung maarte kayo, gamitin natin ang positive result na effervescence or bubble formations. Or, sabi ni Marian, kasi nga nag-aaral na siya for the IELTS, ayaw na niya magtrabaho sa Pilipinas, copious bubbles daw. Okay? Ang arte ni Marian. Alright, let's move on. What precaution is used when performing catalase test? We never do catalase test in the media. Okay? We never do it in the media. We always take a suspension. We always take a strip. We pick out the colonies, and then we perform it in either a tube or a slide. Okay. So the precaution is do not add 3% hydrogen peroxide directly on the blood agar plates, as blood contains catalase. Also, ang dagdag ko lang dito is ang dagdag ko lang dito, class, is when you are doing catalase test, ba't nawala? Anong nangyari? Na-disconnect si na-disconnect ang moderator ko. Siyempre, prepared ako. Kukunin ko din yung wait lang, class. Kukuha lang ko ayan. Ito yung file, open, slide number, something about. Ito ba yung pinag-usapan natin? Do not add, ayan. Yan ba yung natin?
Sorry sir, na-disconnect ako.
Oo nga, nag-disconnect ka eh. Buti na lang nakahabol ka. Kasi mag-ano na sana ako, mag-present na sana ako. Teka lang, anuin ko lang yung presentation ko. Okay na ba ako? Wala na ako na ano. Okay, stop sharing na ako ha. Ikaw na yung mag-present ha. Okay, so don't add hydrogen peroxide. So also in this case, pick out the colonies on the surface ha. Wag yung sobrang lalim nung ano niyo para kayo kumuha ng ice cream ha. Class, yung inoculating loop niyo ginawa niyo pang ano ng ice cream na halukay yung agar ha. Taas lang na part class ha. Wag yung kakantiin yung dugo. Okay? Doon sa blood agar. Wag na kayo magsama ng ng agar. Ang mga microbiologists, dextrose ang mga kamay ng mga yarn. Okay? Arte dextrose. Diba? Alright. Anyways, let's move on to the next slide. Based on anaerobic and aerobic growth, differentiate staph from micrococcus species. So, di ba, pinahirapan ko kayo, babalik din naman pala tayo sa micrococcus versus staph. Micrococcus versus staph aureus, ano sa tingin nyo? Aerobic or anaerobic? Survey. Survey na lang tayo, sige. Sa mga tao tayo. Sige, lahat, sagot. Chat. Chat lahat. Aerobic. Aerobic, isa lang ang sumagot. Si Marian lang ang student ko. Wawa naman yung iba. Pag hindi sumagot, babagsak. Charot. Ganon. O diba nagsisagutan? O diba? Aerobic. O diba nagsagutan? Ang galing ang galing mag-manipulate at mag-gaslight, no? Hindi ako pwedeng magkaroon ng relasyon. Okay, pakita na nga natin ang sagot kung tama ba ang lahat. Oh, okay. So, yon. Facultative aerobe siya. Okay? It can go both on aerobic and anaerobic. Okay? Okay? Grows on aerobic conditions only. Or aerobic, obligate aerobe yan ha, hindi anaerobe. Hindi ko napalitan. Okay? Obligate anaerobe siya. Whilst Staph aureus is facultative anaerobe. So it can grow on both aerobic and anaerobic conditions, while micrococcus can grow only at on aerobic conditions only. Palitan nyo yung anaerobic conditions sa aerobic conditions na kaya. Obligate anaerobe siya. Ay, obligate aerobe. Obligate aerobe. Hindi ko pala napalitan. Okay? O, di ba nagsisagutan yung mga tao? Di ba? Ang hindi sumagot, babagsak. Surveys na lang tayo ngayon kasi nagmamadali tayo. Okay. Drug susceptibility test that can be used to differentiate staph species from micrococcus species. We can use drug testing. We can use antibiotics to differentiate them. Lysostaphine test could be used to differentiate them. Another one is bacitracin. Okay? Lysostaphine, from the name itself, it will destroy staph. It will lyse staph. Therefore, staph would be sensitive. Micrococcus, on the other hand, will be resistant. Bacitracin will have an opposite reaction. Staph species will be resistant to bacitracin, while micrococcus will be sensitive to bacitracin. Okay? Clear tayo? Clear? Okay? Tandaan nyo lang, bacitracin, lysostaphine and bacitracin. Lysostaphine, it lyses staph. Sensitive siya. Huwag na nating alalahanin si micrococcus. Punta na tayo sa bacitracin. Ano nga yung opposite na result ng sensitive? Resistant. So, lagi lang silang opposite pag yung dalawang pinag-uusapan. Alright, let's move on to oxidase tests. Okay? Reagent used in oxidase test is tetramethyl. Wait lang, alala ko pa to. Wait, huwag mo munang i-move. Tandali, kaya ni sir to. Kaya ko to. Tetramethyl paraphenylene diamine dihydrochloride. Tingnan nga natin. Challenge ko para sa sarili ko. Tingnan nga natin. Tetramethyl paraphenylene diamine dihydrochloride. O diba? Memorize pa rin this sir after 2 years na hindi ko tinuro. Diba? Kita nyo, yan yung sinasabi ko sa inyo na kailangan nag-i-stick sa utak ninyo. Okay, class? Modified oxidase test, we just add dimethyl sulfonamide. Okay? So, oxidase test positive. So, sino siya? So, Staph species will have a negative oxidase test, while Micrococcus species will have a positive oxidase test. Ano ang gamit natin? Tetramethyl paraphenylene diamine dihydrochloride. O di ba? Memorize pa ni sir. proud ako yun yung ano. Mamaya meron pang iba class. Chill lang kayo, meron pang iba. Okay, so these are the results. Okay, now let's go on to the metabolism mechanism of the following organisms. So metabolism, are they what? Are they oxidative or are they fermentative? Yun lang yung dapat yung tandaan. So Staphylococcus, because it is capable of living on both oxidation and on both aerobic and anaerobic environments, it is what? It is fermentative. Okay, while Micrococcus species are oxidation-based organisms. Okay, their metabolic patterns are mostly oxidative. Okay, obligate aerobes nga di ba? Okay, class. Okay, now let us try to the test used to differentiate the test used to detect the most important pathogenic determinant of Staphylococcus aureus. This is coagulase test. Okay. There's two types of the test. We have the tube test and the slide coagulase test. What we're detecting with the slide coagulase is a cell-bound coagulase. And what we're detecting with the tube coagulase test is free coagulase. Okay? Clear? Clear tayo, ha? So, I think the difference between slide coagulase and tube coagulase came out of the, what did they call this? The Board of Certification ACP exams. So please be mindful of the difference between the two. Pakitandaan class ko anong pinagkaiba ng tube at slide coagulase, okay? So cell-bound coagulase, cell-bound, slide, tube, free, okay? Alright, now, ito na yung sinasabi ko, nilito ko kanina si Moyo dito. The reagent used in each type of coagulase test. Di ba nilito ko si Moyo? Kumuha ka ng dugo ng rabbit. Di ba sabi ko? Charing lang yon. Kasi ito yon, coagulase ang tinetest natin, Moyo, hindi catalase. Sa catalase, ano ang gamit natin? Hydrogen peroxide, Moyo, ha? Pero sa ilang percent, Moyo, ang gamit natin na hydrogen peroxide? For catalase, bago tayo mag-move on with coagulase muna. Ako nakalimutan na kaagad. Kayo talaga, ha? Oh. Sino makakasagot? Sige, ano na lang. Survey ulit. Sige, class. I-type sa, 1%, 2%, 3%, 4%, 5%. O, ayan na, o, diba? Ang hindi sumagot, babagsak sa board exam. O, diba? Ang galing ko talaga mang-gaslight ng students. Diba? Bakit ko kayo pinagpa-participate, class? Alam nyo kung bakit may tinatawag tayong kinesthetic learning. Kung hindi kayo gumagalaw, hindi nyo ginagalaw yung mga katawan at galamay ninyo, class, baka walang nare-retain. Diba? Okay. So ano na, reagent used in each type of coagulase test. We need rabbit plasma, of course. Next slide, please. Okay. So, slide coagulase requires rabbit plasma or serum. Tube coagulase, you need a specific amount, 0.5 ml. Okay? Tube coagulase. Medyo mas maarte siya. Okay? Alright. We have two types of anticoagulants that you can use, but the preferred one is what? Anticoagulant used for coagulase tests when you're recovering the serum. Or sorry, the plasma. Sorry, not serum. EDTA is preferred. Citrate might cause false positives. Okay? Okay? So, dalawang klase, citrate or EDTA. But we prefer EDTA. Kaya nga ang color ng ano natin, ang color, tandaan nyo yung color, ng background ng slideshow natin. Okay? What is the maximum allowable time to perform tube coagulase? You only have 4 hours to test this. And you have to check it every 30 minutes. Is this important, sir? Yes. You will find out later why. Okay, next slide. Maximum 4 hours. Checked every 30 minutes. So, ilang beses natin siya i-check? twice every hour. So 2 times 4 is 8. 8 times na siyang i-check during the duration of the test. Usually, ang gumagawa niyan sa aming hospital dati, mga intern. Ang sama ng ugali. Oo, kasi ang daming ginagawa ng staff. 'Di ba? Ang gagawin lang, sir, okay po, hindi po nagbe-break apart. Bakit kailangan i-check every 4 hours? Okay? There is a need to do the maximum allowable time for the test because of the fact. I think that's the next question, I think. Tingnan natin. Yeah, why is there a need to perform this procedure or checking it every 30 minutes for 4 hours? Because there might be a false negative due to the presence of breakdown substances like fibrinolysins. So you check it a maximum of 4 hours. Hindi yung after 30 minutes lang. Although some procedures would say after 30 minutes, it always says at the very end, leave the samples for at least 4 hours. Bakit? Kasi we might have false negatives. Okay? Next, let's move on to the next one. Negative coagulase tests are confirmed after how many hours? After 20 hours, and these samples should be stored at 4 degrees Celsius. Okay? It should be stored at 4 degrees Celsius. Next, what may be the cause of false positive results for tube coagulase? This is basically because of the citrate anticoagulant, and the organism is a strain that utilizes the citrate. Okay, what will happen is that there will be a release of calcium, and when there is calcium being released in the reaction, there will be a resulting clot formation. Kasi nga, ano nga ba hematology review tayo, ano ba ang calcium? That is factor 4. Remember, right? Majority of the reactions that you will see, whether it's the intrinsic or the extrinsic pathway, it will always require calcium. Both pathways will always require calcium in between the factor reactions as a cofactor. Okay? Now, let's move on to the next slide. Examples of coagulase positive staph species. There are 4 of them. And I don't think we have to remember most of them, but even if we would, Staph aureus subspecies, Staph aureus. Bakit meron ng subspecies, sir? Kasi molecular studies, meron na tayong molecular technology. Okay? So, lalo na pag gumamit kayo ng BioFire, makakakita kayo ng Staph aureus subspecies Staph aureus. Okay? May makikita kayong mga ganon minsan. So, pag gumamit kayo ng mga molecular study like microarray, may mga subspecies na sila na ilalagay. But for the purpose of our discussion, siyempre mga mga baby medtechs pa lang tayo pang-graduate natin before tayo mag-take ng board exam. What we need to remember is Staph aureus subspecies Staph aureus, Staph intermediate Staph hyacus, and Staph schleiferi, subspecies coagulans. Small letter C yan. Okay? And in order to differentiate that, we do tube coagulase, Bogues-Proskauer, and PYR test. What is PYR? Pyrrolidonyl aryl amidase test. Okay? Ang dami ko na naman. Memorize ko pa rin kung ano yun, di ba? Huwag niyong kakalimutan yung mga ganern. Pyrrolidonyl aryl amidase. Di ba? Mga ganern. Okay, what is the result expected from a coagulase positive staph species? Well, you will see a table, right? Pakita nga natin ang table. Now, what you will notice is that Staph aureus and Staph aureus subspecies aureus and Staph aureus schleiferi subspecies coagulans, they both test positive for Bogues-Proskauer and tube coagulase. But the difference between the two is the PYR test or the pyrrolidonyl aryl amidase test. Why? Because class, one of the deciding factors to see whether or not your organism is Staph aureus is PYR test. Pag sobrang dami ng confusion, PYR test ang ginagamit. Nung unang panahon. Okay, nung wala pa tayo nung mga Vitec natin, Bactec, mga ano natin. What's the other one? API manual, yung mga nilalagyan ng oil. Okay, mga ganun. Tsaka nung mga Phoenix BD na yan. Nung mga wala pa yan. Manual yan ginagawa. Naabutan ko yan, class. Pero hindi pa ako ganun katanda, class. Ha? Huwag kayong ganun. Huwag masyadong bastos, ha? Hindi pa ako ganun katanda. Patrick, ha? Tumatawa ka Patrick. Patrick, hindi pa ako ganun katanda. Naabutan ko lang yun. Kasi nung time na nagtatrabaho na ako, mahirap ang ospital namin. Charet. Ayoko baka mamaya bigla ako sugurin. Charing lang. Okay? So, yan. Bakit hindi mo diniscuss, sir, yung dalawa sa Bayaan mo sila kasi variable sila eh. Okay? Huwag niyong pandaan yan. Variable yan. Kaya hindi ko pa yan nakita lumabas sa board exam. And mind you, I've been doing this since the beginning of time. Charot! Since the beginning of time. Char lang. Okay, let's move on to DNA's test. Alright, so DNA's test may be used to identify Staph aureus and what other organisms can be used. Huwag niyo kakalimutan yung SMS na mnemonic for DNA's test. Huwag niyo kakalimutan ng SMS. Ano nga yung mga organisms na nandun sa SMS na yon? Staph aureus, yung una, syempre. Ano yung isa? Moraxella catarrhalis. And the other one is, what? Other one is Streptococcus pneumoniae. The contents of the media includes, what? Pakita na natin ang sagot. DNA, of course, and methyl green. Okay? DNA and methyl green. Huwag niyo kakalimutan yung SMS, ha? Makakakita kayo ng mga organisms sa ganyan. Although, meron pang iba, pero most of the time, okay, pag ini-etchos kayo ng mga examiner, tatandaan nyo lang sila. Ano ba pinagkaiba ni Staph aureus din sa dalawa? Sige nga. Kung naaalala nyo. First of all, gram-positive siya. Second of all, cocci siya. Diba? So, pag nakita ninyo as case study yan, dapat ganun ang gana ng utak ninyo, class, ha? Oh, papakita doon slide was Sir Marco, may picture ba ako? Pwede ako magbigay ng pictures sa exam ko. Nandiyan ba si Sir Marco? Hindi ko maalala kung may nagbigay ako ng picture. Pero okay, so hindi ko alam kung pwede ako magbigay ng picture. Gusto ko yung mga yung mga nandito sa ospital namin. Para
Yung picture ba?
yung sa exam nila, magbibigay ako ng mga picture sa exam nila.
Kung kaya magdamihan, sir, sige lang.
Ha, manda kayo yung mga picture dito sa ospital gagamitin ko. At hindi kayo makakapalag kasi alam nyo kung bakit. Class, alam nyo kung bakit. Meron kaming molecular analyzer dito, class. Kaya on the molecular level ang ID. Okay, hindi kayo makakapalag. Okay. Bibigay ko pati yung case ng pasyente. Of course, hindi natin i-disclose yung name kasi baka makulong ako. Okay? Kasi maarte dito sa US. May hipa na tinatawag. Anyways, let's continue na. Let's move on to the next slide. What is the action of A protein? Oh, protein A, sorry. Protein A. So protein A is an phagocytic inhibitor. Okay? And its action is it binds to the FC portion of immunoglobulins. Let's see if that's correct. Sinare ko na sa inyo, class, yung kasi nagmamadali tayo. Okay? And I don't like to play with your time. Okay, class? Alright, so it's a phagocytic inhibitor and it naturally absorbs the FC portion of immunoglobulins. Alright? Now, let's move on to the next one. Enzymes released by Staph aureus. There are 5. I-recite ko siya isa-isa. Tingnan natin kung naalala ko pa. Staphylokinase, DNase, hyaluronidase, lipase, and coagulase. Tingnan nga natin kung tama si Sir Manuel. O, diba? Ang function of staphylokinase is a fibrinolysin. It is one that breaks apart the coagulase. Okay? Fibrinolysin. It breaks apart the fibrin. It breaks apart fibrin. DNase breaks apart DNA, obviously. Hyaluronidase is a spreading factor. It breaks apart hyaluronic acid. Lipase, fats, and coagulase, it is the one that initiates clot formation. Okay? So there are 5 toxins also. Huwag niyo kakalimutan, class. Tingnan nga natin kung memorize pa rin din ni Sir Manuel. Exfoliatin, leucocidins, hemolycins, and staphyloenterotoxins. Okay? Exfoliatin, leucocidin, hemolycins, and staphyloenterotoxin. O, di ba? Memorize ko pa. Hindi yan nawala sa utak ko. Mga nakatato na yan. Permanently na yan. Kumbaga, pag in-MRI ako, class, tapos merong yung MRI, makikita nila yung part na yan. Okay? Ganon. Next naman tayo. What is the effect of an exfoliative toxin? It causes Reiter's syndrome, otherwise known as scalded skin syndrome, SSS. Hindi tinatawag nating social security scam. Oy, Char, hindi ganon. Oy, political ka, sir. Wag ganon. Bacteria lang to. Wag ganon. Hindi SSS. Hoy, chill ka lang, sir. SSS is also known as scalded skin syndrome. Yung parang kang nalapnos. Okay? Enterotoxin causes TSS, toxic shock syndrome. What other organism causes toxic shock syndrome? Sige nga, what other organism causes toxic shock syndrome? Patay. Streptococcus pyogenes. Huwag nyo kakalimutan, class, ha? It has enterotoxin. Okay? Mamaya natin babalikan, pupuntahan siya. Okay? Clear na tayo? So, exfoliative toxins, scalded skin syndrome, or Reiter's syndrome. Enterotoxin is toxic shock syndrome. Next, another name for leukocidine is what you call Pontu-Valentin factor. Let's see if that is correct. The Pontu-Valentin factor. Okay? Now, we're done with that. We're done with strep. We're done with staph. We're now going to move on with strep. Medyo mabilis ako, ang bilis ko din magsalita, pero kung gusto niyo ng clarification, sabihin lang, sir, huy, wait lang, chill ka lang. Okay? Ganun kayo sa akin, ha? Chill ka lang, sir. Okay? Naalala niyo lahat, class? Na-retain ba, class? Okay? Okay, mamaya bibigyan natin kayo ng recap. Okay, let's move on to Streptococcus species. Medyo mahirap si Streptococcus kasi there are different ways of identifying the organisms. But what I can help you out with is how they're going to ask this in the board exams. So let's start first with the basics. Classification. This is the classification that is based on the antigenic group specific cell wall polysaccharide. So we're looking at the cell wall itself, the antigen specific groups. Okay? So we have the Landsfeld classification. Diyan yung maririnig yung group A, group B, group C, group D, group E, group G, strep. Okay? Group A ang pinaka-important sa atin as medtechs. Okay? When you see something as group A, lalo na kung nasa blood or nasa may mga throat swab, sabihin na kaagad sa mga lalo na sa mga samples sa sterile site, sabihin kaagad sa senior. Okay? Next, let's talk about the common medically important group B strep. Aside from strep pyogenes, we have what we call Streptococcus viridans. Okay? Group B, Streptococcus viridans. That is usually seen in neonatal infections. Kaya meron tayong makikita sa ospital na GBS swab or GBS screening. Strep viridans yung tinitingnan natin doon, or group B strep. Bakit, class? Yan yung nagkukos ng neonatal infections, lalo na kung hindi na-treat. Also, spontaneous abortions. Okay? Yan yung kinukuhanan si mother ng rectal at vaginoanal swabs, or mga tinatawag na two swab techniques, yung one in the pink, one in the stink. Okay? Ng mga swabs. Yun yung tawag ng mga dito eh. Yun yung mga tawag ng mga dito eh. GBS swabs. Okay? Now, we also have Streptococcus faecalis, now known as Enterococcus faecalis. Okay? Binago na ang name lalo niya. Class, tawag nyo makalimutan, ha? Medyo matagal na panahon na yan. Okay? Actually, before pa akong estudyante, pinalitan na yan ng Enterococcus faecalis. And it's not capital F, sorry. Nagkamali tayo dyan. Is seen as what group of Landsfeld classification? Is group D, also known as the Enterococci family. Okay? Enterococci family. Now, let's move on to the next slide. This is the Streptococcal classification based on hemolytic patterns. Streptococcal classification is known as what you call the Brown classification. We base it on the alpha, beta hemolytic, and gamma hemolytic patterns. Strep pneumoniae and strep viridans is part of, and some group Ds are members of group A, alpha Brown classifications, and the beta Browns is the group A, B, C groups, and some groups. But what is most important for us is strep pyogenes and strep agalactiae. Gamma hemolytic streptococcus are mostly D, your strep faecalis and enterococci, or streptococcus faecalis and streptococcus faecium. Okay? Dalawa siya ha, dalawa class ha. Huwag kakalimutan yung dalawang group D na important sa ating tandaan. Okay? Strep faecalis, Enterococcus faecalis and Enterococcus faecium. Okay? Dalawa sila class. Kasi baka mamaya maloka kayo, okay? Alright? Faecium and faecalis. Magkapamilya sila. Pero mamaya tutulungan ko kayo kung paano sila i-differentiate. But let's start first with Streptococcus pneumoniae and Streptococcus viridans. Para hindi kayo mahirapan, lagi nyo lang ididikit si Streptococcus viridans and Streptococcus pneumoniae. Okay? Pag Brown classification ang pinag-uusapan. Kasi class, ano ba ang color ng viridian? Ano ba yung viridian? Pag Google search nyo kung anong color ng viridian. Si Arian, kasi medyo madilim si Arian eh. Medyo madilim yung screen ni Arian. Arian, harap ka sa amin, tapos Google search mo viridian. Yun nandyan sa tingnan nyo kung anong magiging color ng mukha ni Arian. Diba? Paki-Google search nga. Ikaw din, Claire. Paki-Google search nga. Tapos, i-full screen mo yung color ng viridian. Ayan.
Magiging green po.
Oh, very good. So, idikit nyo lagi si Streptococcus pneumoniae kay Streptococcus viridans. Bakit? Viridian. Ibig sabihin kulay green. Ano ba ang nasa alpha hemolysis? Di ba green? Oh, di ba? Kulay green siya. Ayan, nakikita ko na yung reflection sa mukha ni Claire, oh. Color green. Di ba? So, wag nyo paghiwalayin silang dalawa. Mamaya natin sila paghiwalayin. Beta hemolytic, tandaan nyo lang, Streptococcus pyogenes and Streptococcus agalactiae. Marami akong students nagkakalito-lito sa kanilang apat. Kasi most of the time, our board examiners, they will ask about these 4 organisms. Strep pneumoniae, Strep viridans, Strep pyogenes, and Strep agalactiae. They are all medically important. Very rarely ko lang nakikita sa mga exams yung Strep faecalis, yung Enterococcus species. Okay? So, clear tayo? Tatandaan nyo lang sa kanila, pag Brown classification, lagi ko lang pagdidikitin, Strep pneumoniae. Kasi sinerch ni Claire gonoviridian, kulay green. Diba? Ano ba ang hemolytic pattern na green? Incomplete hemolysis, MHB, methemoglobin, therefore, kulay green yung surrounding area. Okay? Alpha hemolytic yun. Diba? Hindi na kayo nahirapan. May neural connection sa tayo. Alright, let's move on to the next slide. Based on gram staining morphology, describe the appearance of Streptococcus pneumoniae. Bullet shape, flame shape, or lancet shape. Okay? Mukha ba siyang bullet shape? Mukha siyang bullets, di ba? I don't see the lancet and the I don't see the lance or the flames. By the way, class, this is from a blood this is from a blood culture. Okay? This is from a blood culture. Okay? Galing yan sa dugo ng pasyente. Okay? Lancet shape, bullet shape, or flame shape. Okay? Next, let's talk about colonial morphology naman. Colonial morphology is yung appearance niya as colonies on the blood agar plates. Describe the colonial morphology of Streptococcus species. They have what you call a pinpoint colony with a characteristic hemolytic pattern depending on species. Okay? So, pakita nga natin. Ang sagot, pinpoint colonies. Alright, next, let's move on to the next slide. There are 2 medically important beta hemolytic Streptococcus that is commonly identified in the lab. What are these? This is Strep pyogenes and Streptococcus agalactiae. Streptococcus pyogenes will test positive for PYRAs and bacitracin sensitivity. PYRAs, ano nga yung PYRAs? Pyrrolidonyl aryl amidase test. O diba, memorize pa rin talaga dito. Kasi paulit-ulit yan nung aral ako eh. Hindi talaga siya nawala sa utak ko. Pyrrolidonyl aryl amidase and bacitracin sensitivity. Opposite results will be seen for Streptococcus agalactiae and Streptococcus agalactiae. Okay? PYRAs and bacitracin sensitivity. Now, for CAP test and hypurate hydrolysis, what we are looking for is Streptococcus agalactiae to test positive. Okay? Opposite results for Streptococcus pyogenes. Okay? Pero hindi ko na pinahirapan yung sarili niyo and giving you guys a chart. Kasi class, opposite results yan. Okay? Ayoko kayo mahirapan, class. Pag sinabing what tests are used to differentiate chained tawag dito, chained gram stain cocci or gram positive cocci in chains isolated from blood or the throat, ganon. Isolated from a blood culture of a 37-year-old male. Diba? Bibigay doon yung ano niya, yung medical history, and then inilabas dito. Positive, this organism tested positive for PYRAs, positive for bacitracin sensitivity, or it only said sensitive to bacitracin, negative for hydrurate hydrolysis, and negative for Christie-Atkins, Munch, and Peterson test. Diba? Ini-etos lang kayo, ganon. Diba? Okay? So yun yung yun yung easy way to remember it. Next, we go to PYRA's test. Test used for beta hemolytic streptococcus test. In PYR test, which beta hemolytic streptococcus will test positive. As I mentioned before, it is Streptococcus pyogenes. The substrate used in the test is pyrrolidinyl aryl amidase. Okay? The substrate. The color developer, wag mo muna pipindutin. Memorize ko yan. Wait. Chill ka lang. Wait. Memorize ko yan. Para dimethyl aminosinimaldehyde. Tama ba? Pakita nga, ang positive color result is red. Oh, di ba? Memorize pa talaga ni Sir Manuel. Grabe naman hanggang ngayon. Memorize mo pa yan, sir. Over ka, sir. Para dimethyl aminosinimaldehyde. Okay? Saan galing yan? Sa cinnamon yan, class. Di ba? Paano ko naaalala kasi merong mga neural connections. Mahilig ako mag-bake. I use cinnamon. Ay, ito yung ginagamit for PYR. Di ba? Ganun, class, para hindi mo wala. Gamitin nyo in your everyday lives. Di ba, class? Alright, let's move on to the next one. Let's talk about the reaction that is catalyzed by PYRs. Medyo may konting chemistry tayo dito, class. Para meron kayong full picture kung ano ba mga pinagsasabi ko. When you're doing PYRA's test or the PYR test, you will see this reaction. That one. Okay? So, yan ang hindi ko alam. Hindi ko na memorize yan, class. Basta alam ko lang mapagigitna si PYRAs. Okay? Si pyrrolidinyl aryl amidase. Pagigitnaan niya yan. And isi-separate niya si pyrrolidinone at si beta-naphthylamide, which will react with our color indicator. Ano nga yung ating color indicator? Para dimethyl cinemaldehyde. Okay? Cinemaldehyde. To produce what type of reaction? What color? Anong color, class? Pakitype na sa chat kung anong color. Baka may magsagot ng green, ha? Sasakalin ko pa may nagsagot ng green. Ang hindi sumagot kung ano ang positive result ng PYR, babagsak sa exam ni Sir Marco. O, di ba? Ang galing-galing talaga. Ang galing talaga mag-ano. Red, o, di ba? Di ba? Ang galing-galing talaga ng mga students. Ang saya talaga mang-gaslight ng mga students. Alright, very good. So yung L-pyrrolidonyl and beta-naphthylamide, it will be separated by PYRAs and it will have two chemicals, L-pyrrolidone and beta-naphthylamide. Beta-naphthylamide will be reacted with para-dimethyl cinemaldehyde to produce a positive color, which is the positive result for the test. Let's talk about CAMP naman. Transcribe CAMP. CAMP is a name of 4 scientists who developed the test. Christie-Atkins, Munch, and Peterson. Okay? We used a test organism here. Yung reagent organism. And our reagent organism is Staphylococcus aureus. Staphylococcus aureus will be intersected with your beta hemolytic strep, and if there is beta hemolytic strep there, such as your Streptococcus agalactiae, it will produce an arrowhead hemolysis. O diba? Ibig sabihin naglalabanan sila, class. Okay? Yun yung na-develop ng CAMP test. Naglalabanan sila ng hemolysins. Okay? Kaya ganyan ang nangyayari. Okay? Clear? Clear, class? Napansin nyo, class? O, tinan nyo sa yung test organism natin is Staphylococcus aureus. Saan siya dyan? Siya yung nasa may x-axis or yung nasa y-axis? Naku, sir, bakit may ano? Bakit may geometry? Akala ko bacteriology to, sir. May x and y axis. Yung horizontal line tayo. Horizontal line is your control organism or your reagent organism. Staphylococcus aureus. And then your your test organism is your patient sample, of course, or the colony is isolated from your patient sample, which is presumptively Streptococcus agalactiae. And if you test positive, it will form an arrowhead colony. Okay? What is the name of the test? Christie, Atkins, and Munch. Okay? So this is it. This is what it looks like. Okay? All right. Let's move on to the next slide, which is sorry. Reagents used okay, memorize ko pa to. Sandali, try natin. Baka mamaya magkamali ako. Reagents used for hypurate hydrolysis, we have sodium hypurate and ninhydrin solution. Positive result is purple. Tingnan natin. Okay, purple color. Tama pa rin tayo. So, sodium hypurate and ninhydrin solution. Alright, the positive result is the purple color. Okay, the organism that will test positive is, of course, Streptococcus agalactiae. Okay? Medyo tinatamad na ako mag-type niyan. Strep agalactiae na lang, di ba? Nahirapan na, tinamad na si sir. Okay? What are the end products of hypurate hydrolysis? So there are two end products that you will get. You will have benzoic acid and glycine. Okay? This will then be the ones that will be tested. Now, we're talking about a CAMP test, Christie, Atkins, Munch, and Peterson test. Okay? That's the next one. Oh, diba? Okay. And what's useful for what it's used for is for Streptococcus agalactiae versus Streptococcus pyogenes, right? We also have what we call reverse CAMP test. Reverse CAMP test is used to detect Clostridium perfringens. Okay? When you hear the term reverse CAMP, what you're testing for is Clostridium perfringens. And the organism that you're going to use this time is Streptococcus agalactiae. Kaya nga reverse CAMP. Okay? Yung dating tinitest, siya na ngayon ang hinahanap. O diba? Ay siya na ngayon ang ginagamit. Diba? Parang palabas yan dati by Nora Honor. Ang dating ginagamit ngayon ay nanggagamit. Parang ano, no, drama-rama sa hapon na ano, na palabas, no? Class? Okay? Next, let's move on to the next slide, which is sulfomethoxazole trimethoprim sensitivity test is used to differentiate what Lancefield group of streptococci. Naku, pinahihirapan kayo, class. SXT lang naman yan. Ano ba yan, sir? Bakit ako pinahihirapan? But it is useful for differentiating group C streptococcus. And that's basically it. We're not going to delve deep into that. We are mostly focused on group A, group B, and group D strep in clinical microbiology. Okay? Now let's move on to the next slide, which has something to do with scarlet fever. Okay? This has something to do with scarlet fever. There are two tests for scarlet fever. First is Dick's test, and the other one is Schultz-Charlton test. Wag puro kabastusan. Hoy, nakakaloka kayong dalawa, Moyo, at saka ni Patrick, puro kayo kabastusan. Hoy, Dick's test is used for based on susceptibility. Okay? Schultz-Charlton is used for neutralization. Puro kayo kabastusan. Nakikita ko yung eyebrows ni ngumingiti-ngiti, si Moyo, tsaka si Patrick kahit nakamaskara, ha? Puro kayo kalokohan. Dick's test, okay? Next, let's move on. Baka tayo ma-demonetize. Huwag kang tatawa-tawa kasi Patrick. Baka ma-demonetize tayo. Charing lang. Alright, let's move on to the next test. The next slide, I mean. Which of the two sets of tests for scarlet fever is diagnostic? Actually, the first Dick's test is Schultz-Charlton is diagnostic, right? We can see that in our next slide. Yeah, there we go. And the positive result is the blanching phenomenon. Because what are you testing? You are testing the antitoxin, right? You're using antitoxin, right? So basically, if your patient is presumptively or patient presumptively has scarlet fever, the doctor thinks it is scarlet fever. What they're going to do is inject the small site of the body. I think it's the forearm. I haven't looked at the actual test, but what they will do is it's like your PPD test, but what you will get is blanching phenomenon. Okay, parang namumuti. Okay. And then Dick's test, you will get you're testing for the erythrogenic toxin, and the positive result is rashes. Okay. Now, let's move on to the next one. These are infections associated with Streptococcus pyogenes. You'll have 4. Okay, sore throat, skin infection, and scarlet fever, and post-streptococcal infections. You have to remember the 2 post-streptococcal infections. This includes sequela, by the way, which is I think the next slide. Right. Okay. Sequela. Okay, so post-streptococcal infections could be associated with rheumatic fever, and acute glomerulonephritis. Okay, those are the 2 things that you need to remember. Okay, for example, you were exposed to a Streptococcus pyogenes in the past. You are susceptible to having rheumatic fever and glomerulonephritis. What happens? What happens in glomerulonephritis or acute glomerulonephritis? What happens in this one is that there is antibody-antigen complexes that will form from cross-reacting cells in your body that is supposed to be directed towards Streptococcus pyogenes, but because you've already killed the organism with antibiotics, you don't have the organisms. But because you have certain cells in the body that resemble the appearance or that antigenically resembles that of Streptococcus, the antibodies produced against these organisms will not target the cells because they're not there anymore, but will target parts of the human body or parts of the human body. That's why kaya kanya paulit-ulit ko sinasabi rheumatic fever, acute glomerulonephritis, di ba? So yun. Okay? Next, pathogenic determinants is the next one, I think. Yes. So there are 6 pathogenic determinants that we need to remember. Remember your pathogenic determinants. Remove one of them, they become less pathogenic. These organisms become less pathogenic. Remember? So there are 6 of them: hemolysins, streptokinase, streptodomase, diphosphatidylidine, dinucleotidase, erythrogenic toxin, polymerases like DNase and RNases, and the M protein. Let's see if I'm correct. Pinost na pala ni inunahan na ako ni Patrick. Bida-bida si Patrick. Nagbibida ako, binidahan ako ni Patrick. Pero chill lang. Yung pinakamahirap lang na tandaan yun doon, phosphatidylidine dinucleotidase. Okay? Yun. Okay? So, what do we need to remember? Streptokinase digests fibrin. Streptodomase is the one that clumps platelets. The one that kills WBCs is diphosphatidylidine dinucleotidase, and erythrogenic toxin causes redness in scarlet fever. And the polymerases are the ones that your polymerases or your anti-DNases are your anti-nucleic acidases or enzymes are your DNase and RNases, because they break down those substances and M protein. Right? Now, we discussed M protein earlier. We are going to delve deep into L protein right now. What are the actions of M protein? Right? Correct? So the actions of M protein are as follows. It clumps platelets, it prevents WBC chemotaxis, it prevents phagocytosis, and it has a fibrinogen-like action. Okay? Okay? So, it has something to do with clumping generally. Okay? And that's basically what it does. Now, let's move on to the next one. Hemolysins produced by Streptococcus pyogenes can be either of the two, streptolysin O or streptolysin S. Streptolysin O is O2 labile. Huwag niyo kakalimutan, O2 labile, Sol. Okay? Sol is your streptolysin O. SOS is your Streptococcus stable. O2 stable streptolysin. Okay? Huwag niyo kakalimutan na sol and sos. Yan yung ano ko dyan. Yan yung mnemonic ko dyan. Sol and sos. Streptolysin O is labile. Streptolysin S is stable. Okay? Sol and sos. Next, hemolysins. The hemolysins, the oxygen labile type, are produced by strep, is considered to be toxic to what cells or tissue? It is toxic to both RBCs as well as heart cells or your myocardial cells. Okay? Myocardial cells. Okay? Kaya nga meron tayong rheumatic heart fever. Diba? Class. Alright. Bakit? Ano ba ang target niya? Yung hemoglobin. And what cells aside from the globins pala ang target niya. So what type of cells has globins? Myocardial cells. They have troponins, di ba? It targets globins. Okay? Next, inoculation techniques is I think the next one. Right. Medyo kadiri ito. Inoculation technique done to identify oxygen labile hemolysins, stab and stick method. Okay? SLO or oxygen labile strain or streptolysin O lyses blood in the stab area. Okay? Sorry. Streptolysin S is the one that has a hemolysis pattern on the stab area. Streptolysin S in the stab area. Streptolysin O does not lyse the area that has been stabbed. Mali yan. Yan yung question ko dati dun sa mga students ko. Okay? So, let me just type it here for people. SLO, the S, or streptolysin O, labile. Okay. Next, neonatal infections are commonly associated with what beta hemolytic strep? Neonatal infections, kakasabi ko lang kanina. Neonatal infections is your group B strep, Streptococcus agalactiae. Okay? Group B strep, Enterococcus agalactiae. Next slide. Okay, so we're talking about group D strep. Okay, so there are two groups of Streptococcus. We have the Enterococcus group and the non-Enterococcus group, both of which are still family members of the streps. Okay, they are still strep species, but they just have a different antigenic determinant, or your Landsfeld determinants. Enterococcus species includes your Enterococcus faecalis, Enterococcus faecium, Enterococcus durans, Enterococcus avium. Bakit Enterococcus ang basa mo, sir, hindi S? Kasi S na nag-iumpisa. Kasi nowadays, it's now referred to as Enterococcus. Okay? These four organisms are now Enterococcus. The non-Enterococcus species are now Enterococcus bovis and Enterococcus equinus. Enterococcus bovis has now been named as Enterococcus let me just double check if the spelling is correct. Sorry, not alginolyticus. Remove that one. XXXXX. Not alginolyticus. Streptococcus galolyticus. Yun yung bago niyang name. Strep bovis. Kasi ano na siya, mas marami na siyang members. Streptococcus bovis group na siya ngayon. Okay? And one of the members, the primary members that you need to remember is Streptococcus galolyticus. Mali pala spelling ko. Pakitype ka din dun sa ano para lumabas sa YouTube, Patrick. Nakaano ka, di ba? Nagbabantay ka doon.
Sige po.
Yan. Kasi lumalabas yan kahit na ilang taon na yan na nag-live. Napansin ko lang nung naglalaro ako ng online games. Alright. So, we have two types. Please move the slide na ano. Next slide na tayo before you type. Ayan. So, there are two types of there are two tests. There's a test to differentiate the two group Ds from each other. We have bioesculin test or bioesculin hydrolysis test or your BEA. Okay? Okay, BEA. What is BEA? Using BEA pala. Bioesculin agar, yung BEA. Tawag natin yun. Okay? Okay, ayun, tinype na ni ano. Okay? So, the components of the media, 40% bile and the other one is esculin. Okay? Bioesculin hydrolysis test. Okay, there's two components of the test. Now, let's talk about the reaction occurring in BEA. So in BEA, what's happening is that you get ferric chloride in the medium. Ferric chloride in the medium reacts with esculatin, which is basically the breakdown product to form black precipitate. You would have this one. If there is hydrolysis, okay, if there is hydrolysis, you will see a black color change. Okay? So the ferric chloride in the media reacts with esculatin, which is the breakdown product to form black precipitate. Your control or your negative control is the one on the side. Okay. Next. Next slide, please. Test that may be used to differentiate enterococci from non-enterococci species is there's two tests: growth on 6.5% NaCl and PYRAs test. Enterococcus species will test positive for both. Okay? Enterococcus species will test positive for both. So ano nga ba yung mga non-enterococcus species natin? Strep bovis or Galolyticus. And what else is there? Streptococcus equinus. Okay? Alright. Let's move on to alpha hemolytic streptococcus. Okay? I think that's the next topic. Okay? Let's move on to alpha hemolytic streptococcus. So there are examples of alpha hemolytic streptococcus that are of medical importance. We mentioned that one before. Sabi ko kanina, di ba, wag niyong paghiwalayin si Streptococcus pneumoniae at Streptococcus viridans. Kasi ano ang color nilang dalawa? Green. Diba? The test to differentiate the two, there are 5. They are, all of these tests are positive for Streptococcus pneumoniae. You test for animal inoculation, you use octochin sensitivity, you use bile solubility again, you use inulin fermentation, and the other one is Neufeld-Kilang test. The only one that will test positive for Neufeld-Kilang is the one with the capsule. Diba? Which one has the capsule? Streptococcus pneumoniae, right? Clear? Clear ba tayo, class? Bakit? Bakit important si Streptococcus pneumoniae? Anong dapat natin tandaan kay Streptococcus pneumoniae? Meron siyang carbohydrate capsular antigen. Kaya siya nagpa-positive with Neufeld-Kilang test. Okay? Clear? Another name for optochin, class? Baka maloka kayo. Anong another name for optochin? Class, who knows the other name for optochin? Taxo A ba or taxo B? Taxo P. Taxo P, o diba? Si Jericho alam, o diba? Ang hindi sumagot, hindi makakapasa sa board exam. Ano nga ulit ang other name for optochin? O ayan, o diba nagsagot dalawa? O yung iba hindi sumasagot, bahala kayo ayan, o diba? Wala, sila lang. Hindi sumagot yung iba. Bahala kayo. Sabi ng kaibigan ko, may gaba daw ako. Takso po. Ano daw yun, Alessandra? Takso po. Ano yung takso po? Kayo talaga, ha? Alright. Okay, so these are the tests. These are the organisms that are alpha hemolytic. And then those are the tests: animal inoculation, optochin sensitivity, bile solubility, inulin fermentation, and Neufeld-Kilang test. Now, let's move on to the next class. Let's move on. Tests used to detect the presence of amidase producing strep. Amidase producing strep. Inartihan lang yan, class, ha? Wag mo munang next, Patrick. Kasi baka hindi ko memorize. Sandali. The test for that is bile solubility. We use sodium desoxycholate. That is the bile salt that is used. The end product of this test is muramic acid, which dissolves colonies. End product includes muramic acid and alanine. Tama ba? Tingnan natin which dissolves the colonies. Positive result is dissolved colonies. Okay, di ba? Sodium desoxycholate ang gamit natin. Okay? O, di ba? Memorize talaga. O, di ba? Nakatato talaga yan sa utak. Ano ba kayo, class? Araw-araw namin ginagawa yan. Nababasa ko lagi yung mga product. Di ba? Okay? Magagawa nyo din yan, class. Okay? Another name for Taxo P is, I think, the next slide. Let me see. Tingnan natin. Taxo P. Okay. Bago natin i-move, tingnan nga natin kung tama ang kung magaling pa rin ang memory ko. Ethylene hydrocuprin hydrochloride. Sige nga. Ethylene diba? Hydrocuprin hydrochloride. Okay? So, huwag nyo kakalimutan, another name for optochin is ethylene hydrocuprin hydrochloride. So, Taxo P is actually what? It is actually copper-based. Paano mo nasabi, sir? Hydrocuprin. Cuprin. Okay? Cuprin is the archaic terminology for copper. All right. Next slide, please. This is the most sensitive serological test. Most sensitive serological test used to detect Streptococcus pneumoniae. We already tested that. We already asked you about that one earlier. That is the Neufeld-Killang test or capsular swelling test. Components of the test include anticapsular antibodies and methylene blue. Okay? Other name for that one is capsular swelling test. Okay? Okay. There's a kit there made by BD. I'm not sure. BD Diagnostics. They call it NF test or NF capsule test or NF cap test. That's the trade name. Hindi ako binayaran ng BD. Okay, class. Wala hindi to paid hindi to paid discussion. Okay? Next, let's move on to the next topic, which is about nutritionally variant streptococcus. This was a recent addition to my lecture. The previous lectures, if you noticed, way back in 2019 below, hindi ko to sinama kasi hindi siya tinatanong sa exams. One time, may nagsabi sa akin na previous student, Sir, isama nyo na yung nutritionally variant streptococci. So, sinama ko na siya ngayon. Another term for nutritionally variant streptococci is what you call your Abiotrophia species. Okay, Abiotrophia species. The growth requirements for this one is that it requires cysteine and pyridoxal. Okay, or vitamin B6. Let's see. There are two species under this genera, which is Streptococcus adjacens and Streptococcus defectivus. Okay? So, Abiotrophia sila, pero Streptococci pa rin sila. Okay? Okay? Pyridoxal or vitamin B6. They require cysteine and vitamin B6. So, if everything else has the tests, pero nakita ninyo yung word na vitamin B6 or pyridoxal, kasi yun yung lumabas sa my student wasn't able to remember the whole question during her ASCP exams. Naalala lang niya yung Abiotrophia, merong vitamin B6. Okay? Always assume it is a nutritional variant Streptococcus. Because one of the choices was Streptococcus pyogenes, Streptococcus bovis /galolyticus, Streptococcus mutans, and then the other one is nutritionally variant Streptococcus yung question. Okay? Choice pala. So, yon. Dapat maalala. Pagka na rin nakita nyo yung vitamin B6 na yan, tsaka yung pyridoxal or cysteine. Cysteine na yan. Sa case. Okay? Next. Dental caries. Streptococcus associated with dental caries. Huwag nyo kakalimutan yung M&M's. Okay, M&Ms. Ano yung M&Ms na yan, sir? Streptococcus mutans, Streptococcus mitis, and Streptococcus sanguinis. Sir, bakit siya important? Kasi class, I've been working in the laboratory under the microbiology department for quite some time now. And one thing that I found is that I would always see these organisms in blood culture, contaminated blood cultures. Ano yung contaminated blood culture, sir? Yung hindi naman siya, ang pasyente is healthy, or the patient doesn't have any pathogens, any bacterial infection in the blood, pero may tumubo dun sa culture, sa blood culture. Yun yung tatawag natin na contaminated blood culture. Paano nangyayari yun, sir? Well, sinabi 'di ba kung saan siya galing? Sa dental caries. Napansin ko kasi most of the nurses kapag nasa nasa mga ICUs na sila, kasi usually yung mga kinukuhanan ng blood cultures yung mga ICU patients, nagchichikahan sila. So, ewan ko ha, proud ako dito. Isa sa mga sinabi ko nung naging infection control, nag-head ako ng infection control infection control link lang, hindi naman head. Pero sa buong laboratory yun. Sabi ko mag-provide tayo ng training sa mga nurses na nagko-collect ng blood for blood culture. Bumaba ang contamination rate namin. Napansin kasi namin, nung wala silang masks during blood collection, mataas yung percentage ng contamination rate, and usually it's Streptococcus mitis and Streptococcus mutans yung tumutubo, 'di ba? So napansin ng boss ko, sabi niya sa akin, anong pwede nating gawin? Baka nagdadaldalan lang yung mga nurse, mahilig yan magchikahan yung mga yan kapag kumukuha ng dugo. So yon, pinagsuot namin sila ng mask for 1 week, tapos tiningnan namin yung the following week, kasi 5 days ang growth ng 5 days ang growth, ang iniiwan sa Bacti-alert ang samples. O diba? Lo and behold, bumaba ang contamination rate. Kasi pala nagchichikahan ng mga nurses habang kumukuha ng blood. O diba? Aside of course, syempre sa pero theory ko lang yon. Madaldala mga nurses. Charit. Shout out sa mga nurses. Friends ko yung mga yan. Alright, let's go to the next slide. Streptococcus, I think that's the one with liver and brain infection. Yeah. Okay, yan na. Okay, so we have three organisms in mind. Please don't forget about Streptococcus sanguinosus, Streptococcus intermedius, and Streptococcus milleri. There are 3 of them: Streptococcus sanguinosus, Streptococcus intermedius, and Streptococcus milleri. Please don't confuse Streptococcus sanguinus or sanguis and sanguinosus. Sanguinosus sa liver, okay, and brain. Sanguis sa dental caries. Okay? Alright, let's move on to the next one. Conditions associated with Streptococcus sanguinosus is your SBE, subacute bacterial endocarditis, can be caused by Streptococcus sanguinosus. And this is going to be our last topic for today. The next one, I think that's the last one for today. Oh no, may isa pa. Siyempre, Neisseria and gram-negative or cocci pa. Other gram-negative cocci. Okay? So, I think punong-puno na ang brains ninyo. So, medyo madali lang si Neisseria and gram-negative cocci. Huwag nyo kakalimutan. Tuturuan ko din kayo mamaya. 10 minutes lang to. I'm sure hindi nyo na makakalimutan ng Neisseria after this class. Okay? So, Neisseria species. Let's start with Neisseria species first. What is the morphological characteristics of most Neisseria species? Morphological characteristic is that they are gram-negative, coffee bean-shaped or kidney bean-shaped diplococci, except for one special member of their family, which is Neisseria elongata. Okay? Let's see if that's correct. Gram-negative coffee bean / kidney bean shaped diplococci or diplococcus, and it is except for Neisseria elongata. Okay? Now, let's talk about the next slide, which has something to do with the morphology. So aside from morphology, how is Neisseria elongata different from the other species of Neisseria based on biochemistry? So we're talking about biochemistry. All Neisseria species are catalase positive, except for Neisseria elongata. Okay? Except for Neisseria elongata. Tandaan lagi And next, aside from that, we are going to talk about the Neisseria species in general. There are two things that you need to remember for Neisseria: catalase and oxidase test. They are both catalase positive and oxidase positive. Okay? Clear? Clear tayo? Paano tayo nagtetest ng catalase? H2O2, 3%. Oxidase test, sir. Paano ginagawa? Gagawa tayo ng, anong gagamitin natin for oxidase test? Yung oxidase strip, sir. Ganun. Okay? Masyado na mahaba, sir. Punong-puno na ang brain cells ko. Alright, let's talk about Neisseria some more. Neisseria species are said to be capnophilic, which means that they require what percentage of CO2? Ilang percent? Alam ko, isa sa mga trabaho ng mga interns, mag-check ng temperature. Ilang percent? Nag-intern ng mga tao dito. Ang hindi makakasagot, magre-repeat internship. Care of Sir Marco. Dadalhin sa Tondo General Hospital. Walang service. Maglalakad lang. O diba? Grabe ako mang-gaslight. Sa Tondo General Hospital, hoy, muntik na ako mag-duty doon nung nasa nursing ako. Kaya nga ako lumipat ng medtech. Ayoko mag-duty doon. Yung college of medtech namin hindi affiliated sa Tondo General. Okay? Madaming mga nursing students doon na nagagahasa. Okay? Bakit? Sino yung nagtaas ng kamay? May nag-duty sa Tondo General. May magko-contest na maganda naman ng buhay niya doon. Nakasurvive naman siya. May nag-duty ba sa Tondo dito? Baka mamaya. Kumakalat-kalat kasi yan yung estudyante pa ako. Sa Tondo daw may nursing student na hold up tapos na rape. Wala naman, wala. Ba't nagtaas ng kamay? May nagtaas ng kamay, naloka tuloy ako. Okay, 5 to 10% ang CO2 ha. Clear? Clear tayo, okay? Next, what is the most important pathologic determinant of Neisseria species? That is, what? The pili or the fimbriae. Okay? The pili or the fimbriae. Alright? Next. Next na tayo, class. Okay. Presumptive test used for the presumptive identification of Neisseria gonorrhoeae. Hala, nakalimutan ko. The superoxide test. The reagent used is superoxole, not superoxide. Superoxole test, the reagent used is 30% hydrogen peroxide. Hydrogen peroxide. Tingin nga, tama ba tayo? Medyo nag-text sa akin, na-distract ako. Ayoko talaga na nadi-distract. Superoxole test. Magkadikit yung class yung oxol at saka yung super. Okay? Superoxole test. Positive result. Tinanong niya yung positive result sa isang previous board exam dyan sa Pilipinas. What's the positive result? Blue. Okay? Blue. Pakilagay ka lang yan, blue, positive result. Kasi yun yung commercially prepared na yan. Mahirap siyang i-prepare kasi kailangan nung gumawa ng color reagent. And I think you can also effervescence din siya. Pero merong may mga type ng test kits. Gumagamit sila ng color indicator. Kasi may iba kasi nalilito sa reaction. Okay? Okay. Next. Let's move on to the next one. Tests that may be used to confirm Neisseria gonorrhoeae. We can use culture or fermentation tests. Okay? Dito na tayo. Tuturuan ko na kayo after nito yung shortcuts ko. Yung tips and tricks ko para hindi kayo malito pagka sinabing Neisseria. Okay? So we do culture and fermentation. Let's see if it's correct. So culture. Okay? Usapan natin muna ang colonial morphology. I think the first thing that we have to look for is the colonial morphology. But let's talk about the STD caused by Neisseria gonorrhoeae first. That is the next slide, I think. Tingin nga. Yes. Gonorrhea leads to what form of infection in males? In males, it can cause purulent urethritis. In females, it can cause cervicitis. I have a question. This was a question, I think, in Apollon, if I'm not mistaken. Which of the following will have will be a diagnostic sample for Neisseria gonorrhoeae without doing culture? Okay? Purulent urethritis with purulent urethritis from males. Pus from a purulent urethritis from a male patient. Female swab with diplococci in cervical sample, both, or none of the above. Culture is still diagnostic. Yun yung lumabas sa question. So purulent sample from a male, cervical scraping from a female, both, or none of this, because cultures are diagnostic. Sige. Which of the following samples are diagnostic? Taray, nagawa sila ng letter, no? A talaga, no? Wala naman ako sinabing A. Pero sige, gawin na natin A, B, C, D. Sige. Ang hindi sumagot kahit hula, babagsak sa lahat ng exam. Di ba may nagsagutan, di ba? Kailangan pala ganun, Patrick, no? Dati hindi ko kayo mapasagot din sa mga ganyan. Tingnan mo si Bay. Sa sobrang niya, excited niya magsagot, sinagot niya S. Di ba? Kita mo kinabahan siya, ayaw niya ng sumpa. Di ba? Di ba? So tama kayo, class, lahat. Males with a characteristic gram stain, a morphology of gram-negative intracellular coffee bean-shaped diplococci is diagnostic of Neisseria gonorrhoeae infection, and most of the time, physicians, when they see this report, they would immediately give antibiotics. Okay? Except of course sa Japan, kasi doon yung nagkaroon ng first case ng super gonorrhea. Okay? Alright, so ano yung super gonorrhea, sir? Yun yung resistant siya sa lahat na in first line ng antibiotics for Neisseria gonorrhoeae. Okay? On which gender would the gram stain... ito na pala yun. Okay? On which gender is penile discharge or yung purulent discharge na yan. Okay? From males. Okay? Females, grabe naman, pati ba naman yung mikrobyo, napaka-sexist. Diba? Grabe pati mga mikrobyo, no? Lagi na lang male ang kinakampihan. Paano tayo mga babae? Diba? Alright, let's move on to the next one. Paano naman kami mga kababaihan, class? Diba? Lagi na lang silang ganyan sa atin. Pati mikrobyo, lalaki ang kinakampihan. Diba? Alright, so let's move on to another infection caused by Neisseria gonorrhoeae. Eye infections acquired by newborns from mothers infected by gonorrhea. This is known as ophthalmia neonatorum. It is prevented by using 1% silver nitrate, also known as what you call Creed's prophylaxis. But this one is a very old, procedure. They don't do this anymore. What they do now is they have antibiotic drops na. Okay? So yung antibiotic drops, again, iba to kaysa sa, sabihin ko na class, iba pa rin sa Japan. Kasi nga nandun yung first case nung super gonorrhea. So, it could be different. It could be a different protocol when, depending on where you live. But most of the time, they have antibiotic drops for the child. Okay? For the child. Alright, now let's move on to meningococcemia. I think that's the next slide. Meningococcemia associated with Neisseria ningitis is transmitted through two things, inhalation of droplets or close contact. Siyempre, droplet siya. Droplet. Okay? Last week, tinext ko si Sir Marco kasi sobrang paranoid ko. Kasi first time ko maka-isolate ng Neisseria gonorrhea. So kinabahan ako. Sabi ko, Sir Marco, should I be worried? First time sa buong career ko kasi naka-isolate ako ng Neisseria gonorrhea. Sa blood culture pa siya. So tinext ko si Sir Marco, should I be worried? Sabi niya, sa BSL ka naman. Buti na lang kahit ganyan ka-troll si Sir Marco sa inyo, no? Mabait pa rin siya sa akin. Sabi niya sa akin, na sa biosafety cabinet mo naman ginawa, di ba? Tsaka nasa PPE ka naman. Okay ka pa. Makakapag-lecture ka pa. Charet. Okay. Alright, class. So next topic naman tayo. Next slide, I mean. Meningitis associated with Neisseria meningitis infections is common in what age group? Children under 3 years of age. Kaya isa pa yun sa mga reason kung bakit hindi na ako nangamba. Okay? Actually, binalikan ko tong slide na to, class. Binalikan ko tong tong lecture na to. Sabi ko, ito ba yung sa mga bata? Okay? Alright, so next, let's move on with the symptomologies naman tayo. Meningitis accompanied with what sign could be presumptive of meningococcemia? This is what you call scattered petechiae, specifically in the back area. Okay, scattered petechiae. And we also have another one. I think it has something to do with hemorrhagic bleeding in the adrenal glands associated with meningococcemia. This is known as your Waterhouse-Friderichsen syndrome. Okay, you will hear of Waterhouse-Friderichsen syndrome when you study coagulation. When you study coagulation in hematology. Okay, so if it says something, a patient was diagnosed with a bleeding disorder, specifically in the abdominal region. Which of the following? He tested positive with his spinal tap or CSF cultures, as well as a blood culture that showed gram-negative rods, and then it showed you a lot of diseases. Select Waterhouse-Friderichsen immediately. Okay? Ganyan sila magtanong. They will give you a whole list of meningitis symptoms, and then they will say the gram stain morphology. They will say the morphological appearance in the gram stain of both the blood culture and the CSF. You'll see Waterhouse, right? Just select. Don't even read the rest of the tests. Just select Waterhouse-Friderichsen. Okay. Next, biotypes of meningitis that mainly causes meningococcemia. There are 5 things that you need to remember: A, B, C, Y, and W. Okay. So yun yung mga biotypes: A, B, C, Y, and W. Alright, let's move on to the next one. Kasi malapit na matapos ang ating time together. Specimens that are useful for the diagnosis of meningococcemia. There are 3. Okay, CSF blood and nasopharyngeal swabs. Okay, CSF blood and nasopharyngeal swabs. So CSF and blood, they are usually for patients who have actually shown symptoms of the disease. Nasopharyngeal swabs are for patients who have who need to be screened. Like for example, katulad ko. Okay? Naka-isolate ako nung araw na yon. So dapat kinuhanan ako ng nasopharyngeal swab. But because maarte kami, meron kami PPE and everything. Kere bam lang. Pero sinunog ko na. Tinapon ko na yung damit ko na yun. Tinapon ko na yung damit ko that day. Yung scrub suit ko that day. Anyways, let's move on to next slide. Now, we're culturing the organism now. We talked about the disease and everything else, the transmission and stuff about it when it comes to the disease and symptomology. Now, we're going to go with the lab work that is used for us. So, media used for the isolation of Neisseria species, there's 4, but you can use only one of them, and there is antibiotics in each of them. Let's go one by one with Thayer-Martin, modified Thayer-Martin. Martin Lewis and the New York City Algar. Can you show them the that slide? Okay, all of them have the 3 have the combination of the 3 of at least have 2 of them. All of them have 2 of them have 2 antibiotics in common. They have vancomycin and colistin on everything, except for the modified Thayer-Martin, which has nystatin and trimethoprim. So don't forget Stearmartin and modified Stearmartin. The only difference between the two is that there's trimethoprim. They basically have the all antibiotics except for trimethoprim, which can only be found with MTM. We go to Martin Lewis that has vancomycin, colistin, trimethoprim, and anisomycin. Martin Lewis is almost always the same as NYC agar, but the only difference is that instead of anisomycin, it has amphotericin. Amphotericin B to be specific. Okay? So I want you guys to always put them together. So Stearmartin and modified Stearmartin, they're all together. Martin Lewis and the other one, if you don't remember, Martin Lewis and the other one, they're together. What's common between them? Vancomycin, colistin, and nystatin. Okay? What's the difference between why is it modified? Well, they added trimethoprim. Let's move on to the next two. Martin Lewis has the same, vancomycin and colistin, and trimethoprim. But what makes them different with each other is that Martin Lewis has anisomycin. The NYC agar has amphotericin. Okay? Okay, para hindi nyo makalimutan, class, NM. NM and MN and MN. Ano yun, class? Para hindi nyo makalimutan. Martin Lewis, anisomycin. Diba? MN. And then, New York agar, NM. New York City agar, AM, photericin. O, diba? Para hindi na kayo makalimutan. Para hindi nyo na makalimutan. Diba? MN. Babalikan nyo lang sila lagi. Pagbaliktad nyo lang yung letters. Diba? All of them have vancomycin and colistin in the preparation. Alright, now, ito yung para hindi nyo makakalimutan ang mga Neisseria natin. Let's go to the biochemical testing. Most important thing that you need to remember is the sugar fermentation tests. Huwag nyo kakalimutan ang GML. Okay, class? GML. Gagawa kayo ng GML. GML. Okay? Tapos, gagawa ulit kayo ng G. Pababa. M. L. Okay? Anong purpose, sir? Nakita niyo yun na parang hagdanan lang. Okay? Huwag niyo kakalimutan yung yung arrangement ko. Para hindi niyo makalimutan. Nagamit ko yan sa exams ko, class. Promise, nagamit ko siya sa exams ko. Parang hagdanan lang siya. Tandaan niyo na lahat sila Neisseria. So, ang titingnan niyo yung mga pangalan nila, GML. Okay? GML din siya dito sa sugar fermentation tests niya. Tapos, positive lang kay Gono. Tingnan niyo. Parang hagdanan lang, di ba, class? Huwag niyo papalitan yung yung position niya. 'Di ba? Gagawa ka lang ng hagdanan. Kaya ganyan yung ginawa ko na sa chat box natin. 'Di ba? Nagamit ko yun sa board exam, promise. Question sa akin is what is the difference between Neisseria gonorrhoeae in terms of sugar fermentation tests? Dinictate doon. Neisseria gonorrhoeae is positive for glucose fermentation but not for lactose and maltose. Neisseria gonorrhoeae is positive for glucose and maltose. Neisseria gonorrhoeae is positive for all sugar fermentation tests including another one. Mga ganong kaartihan pero isa lang siya. So nagamit ko siya sa exam. Ito yung mga tinatanong, high yield to, tinatanong talaga siya, class. Okay? So kung meron man kayong tandaan yun sa last part ng discussion natin, ito. Okay? So, paano tayo mag-start niyan? Paano tayo, class, niyan? Teka lang, tingnan muna natin kung tapos. Next slide muna tayo. Okay? So, ganyan siya, ha, class. Huwag niyo kalimutan yung arrangement pati, ha? Okay? So, to confirm the identification of Neisseria lactamica, what is done to the sugar fermentation tests? What we do is we do ONPG, ONPG test, which we will discuss in Enterobacteriaceae. Okay, we do ONPG test. Let's go on to the next slide para bibigyan ko kayo ng algorithm to identify the Neisseria species. Okay? Next slide na tayo. ONPG yung sagot dyan. Nasaan na si Patrick? Ano nangyari kay Patrick? Ba't hindi niya ginagalaw? Alright, ONPG. Okay. Next slide na tayo. What base media may be used in testing for sugar fermentation of Neisseria species? We can use a peptone broth or a cysteine trypticase agar. Okay? Cysteine trypticase agar. Dalawa lang siya. Peptone broth or cysteine trypticase agar. Okay? Then dun mo na idadagdag. Diyan mo na idadagdag yung either sugar, maltose, or lactose. Okay? If you're testing for these organisms. Mahirap siya nung nagtitrain ako dati kasi wala kami yung commercially prepared. Kami yung gagawa. So, mag-sterilize ka muna ng peptone broth. Gagawa muna kami ng peptone broth tsaka ng peptone broth, or I think we were using the peptone broth. And then after that one, saka namin idadagdag yung mga yung mga sugars namin. Okay? So medyo toxic siya. Okay? So next tayo. Next is your most culture media. Most culture media used in the identification of Neisseria species is basically what enrichment media. It's basically chocolate agar plates. And aside from antibiotics, what other components may be present in this media? We usually add isovitalex and hemoglobin. Okay? Isovitalex or hemoglobin. That is, isovitalex is a trade name for most BD prepared BD ba siya? Yeah, again, hindi ako binayaran ni BD para sa lecture na to. Okay, class. Wala tong commercial, wala tong ads, okay? And the hemoglobin. So isovitalex TM nga dapat yan, and hemoglobin. Okay? Next. Let's move on to the next one. Genital mycoplasma may be cultivated in what media? We can use it we can cultivate it also with using NYC medium. Dinagdag ko lang siya just for your info. Okay, class? Next. This is a Neisseria species that has the same sugar fermentation characteristic as Neisseria meningitidis. How is the species different in terms of growth requirements? So Neisseria meningitidis, same fermentation. Ano nga yung same? Pag sinabing fermentation, sugar fermentation reaction, ano siya? For like GML, so titingnan natin. Second tier, positive for glucose, positive for maltose, negative for lactose, kasi nga hagdanan, di ba? Okay? So, sino siya? Sino yung sagot dito? Ang sagot dito is Neisseria subflava. It has the same sugar fermentation result but can grow in 25 degrees Celsius. Okay? Siya lang yung Neisseria na capable of growing at 25 degrees Celsius. Okay? Subflava. Okay? Next, we have another one. This is less fastidious. Gram-negative organisms that can grow at 5% on 5% sheep blood agar. We have Neisseria meningitidis, saprophytic Neisseria, and Moraxella. Most fastidious is Neisseria gonorrhoeae because it is an intracellular parasite. Okay? These guys can grow and they can grow happily. I would say they can grow happily in artificially prepared medium. Okay? Okay, next. Gram-negative cocci. I think that's the gram-negative cocci that is seen in lower respiratory tract infection. This organism usually targets what age group? Moraxella catarrhalis. Okay? And this organism usually targets teens. Okay? Next slide. Ay! Wala yung sagot sa may teens. Basta teens siya. Okay? Adolescents. Okay? Let's move on to that one. Biochemical tests used to identify Moraxella catarrhalis from organism. from other from other gram-negative organisms, you would have to test DNAs and sugar fermentation test. It will result to have to negative result on everything. Okay? Kaya hindi ko pinapakalimot sa inyo yung DNA test. Ano yung SMS natin, class? Sino yung SMS? Ano yung shortcut natin? Sino makakasagot? Ang makakasagot sa akin, papasa ng board exam. Makakapag-fly to the United States sa 2027. Oh, grabe naman yung sumpa ni Sir Manuel. Maganda yung sumpa na yun. Walang sasagot. Ano yung ibig sabihin? Anong ibig sabihin ko pag SMS? SMS na related sa DNAs. Oh, kita nyo makakapunta na ng United States yan si Aiza Tolentino. Diba? Si Reisha Roxella ano na naman tayo Aiza eh. Diyan na naman tayo sa kulang-kulang eh. Tandaan mo meron tayong si Reisha Rubidea at saka si Reisha Liquefacients. Charing lang, okay? Pakiayos, pakikumpleto ha, class. Kailangan specific tayo, okay? DNA test and sugar fermentation test is the test that we use for differentiating it from other gram-negative organisms. Okay? Next. Taray, alam na ni Patrick kung kailan ako magsasabi ng next. Inunahan ako. Or minamadali ako. All medically relevant gram-negative cocci are aerobic except for the following. Except for what? Except for Veillonella and Moraxiella. Okay? Okay? Moraxiella. Veillonella and Moraxiella. Okay. Now, we move on to the next one, which we are going to talk about Veillonella species. Okay, Veillonella species, what's the one that's important to us? Veillonella parvula. Veillonella ferments lactose. Okay, it ferments lactose, just so you know. Okay, and Veillonella parvula is rarely associated to human infections. I think that's the next one. Yeah. Okay, but it may cause infections like osteomyelitis and endocarditis. Okay? Okay? And that's basically it. I think that's the end of our lecture, right? Wala na tayong iba? So class, anong naiintindihan natin today? We have 10 minutes. I'll help you guys out with an algorithm para hindi kayo maluka, class. Okay? So every year class, meron akong binabago na sa lecture ko para na truly yours lang class or for your batch only na sandali, lilipat tayo ng okay. Alright. Tapos makikisali ako sa Zoom. Okay. Para hindi nyo makakalimutan at the end of this lecture, nakatato na din sa utak ninyo yung mga Neisseria species. Okay? Paki-accept po. Kung sino man yung nag-a-accept. Ay! Sorry. Okay. Sorry, nag-echo. Yan. Wala na yung echo class. Wala
na po.
Okay. Alright. So, ako naman yung mag-present. Okay? Teka lang. Baka may makita ang mga students na hindi ka nais. I-ready na muna natin dun sa may dun sa talagang kailangan. Share screen. Start broadcast. Tapos yan. Okay? So, start tayo with Neisseria. Kasi meron pa tayong 12 minutes. So, diba 10 minutes lang maalala ninyo yan, class? Anong color ng pen ko? Siyempre, gram negative para hindi nyo maano. Koksay tayo, class. Ay, pangit naman naman. Ayan. Okay. So, let's start with gram-negative koksay. Gram-negative koksay, how is it different? We always start with gram stain, di ba, class? Yun yung sinabi ko sa inyo. We always start with gram stain. Gram positive or gram neg? X na natin to, ibang discussion yan. Gram neg tayo, di ba? So, what's the thing that we do first? Sige nga. Gram stain. What's the thing that we do first? We do what? Catalase test and oxidase test, di ba? So, sino ang magka-catalase positive? Sino ang mag-oxidase negative? Kalimutan na nila. Neisseria ba or yung others? Pakisagot? Sino nagtaas ng kamay? Hindi ko nakita, sorry. Katingin ako sa baba. Taas mo ulit kamay mo. Ayan, si Marian. Oh, sige. Hindi ko nakita, sorry. Okay, go.
Sir, yung tanong po ulit, tama po ba na yung ano yung magpa-positive sa catalase test?
At saka oxidase.
Okay po.
Oo.
Sa positive test, ay, sige po, ano po?
Ayan na, nag-drawing na ako. Sige, sabihin mo sa akin.
Ay, yung magpa-positive po sa catalase test, si Staphylococcus, tsaka si...
Hoy, hindi. Hoy, gram negative kaya nga red yung color.
Gram negative.
Kaya nga lipstick yung gamit ko na pansulat. O?
Wait lang, nalito ako, sir.
Hoy. Ayun talaga. Ibabalik ba natin, Patrick? Yung ano, balik tayo sa umpisa. Nalito siya, oh. Hoy, gram negative tayo. Ito talaga.
Ano, sir?
Oh, ano? Nakalimot na ang mga students. Kayo talaga. Oh, sino mag-gram positive? Sino mag-oxidase positive at magka-catalase positive?
Catalase positive, sir, mga Neisseria.
Okay, Neisseria species, di ba? Ay.
Except yung elongata version.
Oh, di ba? Oh. Species except and elongata, di ba? Correct? Di ba? Tama tayo? Tama, di ba?
Yes, sir.
Okay. Now, how do we differentiate the Neisseria species? Kalimutan nyo na. GML. GML. Diba? Hagdanan lang yan, class. Diba? Diba, class? Kalimutan nyo? Gonorrhea, meningitis, lactamica. Diba? So, ano na nandito sa gilid? Ano yan nandito sa gilid? Na dalawa. Anong dapat nating tandaan? Veillonella. Nahirapan ako. Ang pangit ng ballpen na gamit ko. Parvula. And Moraxella. Anong difference nila? Moraxella cataralis. Anong difference nilang dalawa? Class. Ang difference. Red fluorescence, di ba? then simula kasi ano, DNAs. Oh. Diba? Tapos. Diba, class? Tapos, pahirapan pa nyo sarili nyo, Neisseria subflava. Okay? Neisseria subflava, same with Neisseria meningitidis. But, what's the difference? Grows only at 25 degrees Celsius. O, di ba may policy and procedure na kayo sa gram-negative? Oh, di ba? Pwede na kayong mag-test na Neisseria. Which one is diagnostic for males? Neisseria, gonorrhea, penile sample sa... Okay? Okay? Di ba? Ang haba, ang dami mong chika, sir. Yun lang naman pala ang kailangan namin tandaan. Ang dami mong chika. Ganun lang pala yun. Tapos na pala dapat tayo, sir. Kanina pa pala dapat kami tapos. Di ba? Pero for gram negatives yan. Meron din akong shortcut dun sa last year. Kaya kailangan nyo panoorin yung mga last year. Charet! Scam na naman ako. Okay. So I think hanggang dito lang tayo, di ba? Anong oras na ba sa Philippines? Siri, what time is it in the Philippines?
2:55 po.
Oh, tama-tama, di ba? Hanggang 3 lang tayo. Hanggang 3 lang ba tayo? Hindi ko maalala kung ano ba sinabi ni Sir Marco. Kasi naman gabi-gabi na yun. Anong oras nga ba yun? Anyways, I think that's it. I'll see you guys next week. Same time, same place. Hopefully you guys learned something from me today. If you didn't learn anything, if you learned something today, please send a like naman or a heart. O diba? Ang itim ng kamay. A heart na lang. Send a heart naman. O diba? Okay. Do we have any questions for people here in the in our group chat or group meeting? Any questions?
Sir, anong app daw yung ginamit niyong pang-drawing?
Ay, paid up yan. Talagang pinagyabang ang yabang. Charit. Paid up yan. Ang pangalan niya is Procreate. Ganun. Okay. Ano pa? Aside sa mga kalokohan nila. Ano pa? Meron pa tayong 4 minutes class. Ano pa? Wala nang may questions, class?
Wala na, sir.
Oh, si Amanda talaga. Amanda, nandito ka din pala, Amanda. Buhay ka pa pala, Amanda. Buhay ka pa pala, Amanda. Shoot at this life. Nandiyan pa pala si Amanda. Okay, Sir Marco, I think it's time for me to go. I have a flight to catch later. So, I hope you guys have a wonderful week. I'll see you guys next week. And please, every year sinasabi namin ito ni Sir Marco, do not study 2 days before the exam. Okay? Especially for micro. Okay? Kahit kami ni Sir Marco, hindi namin kaya yun. The 2 days before the exam. How much more pa students na mga suki card, charit. Ang sama ko talaga. Ang sama ko talaga. Charit lang. Hopefully, this is our last lecture na makikita ko kayo pati. Alright? And final notes, be kind to one another. Huwag masyadong bully. Ako lang dapat ang bully. Alright? Bye!
Paalam, sir. Safe flight po.
Paalam.